Okay, this is going to be fairly informal. I've been recovering from a bit of a cold, so usually I prepare a bit of a script when I give a presentation. I haven't had the chance to do that, so we'll just see how it goes. My name, as Chris said, is David Eccles. I work mostly from the Milligan Institute, but I work mostly on my own. The Milligan Institute is this wonderful place at the top of the hill in the middle of Wellington. You can see the harbour here, and it's actually sited within the grounds of Victoria University. Anyway, what I want to talk about today is the Oxford nanopore MinION. Now, the title that I've given for this presentation is New Data, New Analyses. And the newest data that I know of that other people have produced hasn't actually been released yet, is stuff that's been produced on the International Space Station. A couple of weeks ago, a MinION was, well, a couple of months ago, a MinION was sent up to the International Space Station, and a couple of weeks ago, it was actually tested and used. This is Kate Rubins here. I believe this is just before her very first MinION sequencing run. The little smirk on her face sort of, is this going to work? It's a really weird device. But this sequencing run actually did work. This will change. And there we have Happy Kate. After a sequencing run, I think in this case it produced about 250 megabases of DNA. On the International Space Station, they did 4 runs. They did 3 6-hour runs and 1 48-hour run. And I can't quite remember how many total reads that they got, but I know that it was at least over 1 billion bases. So they went from sequencing 0 bases in space to 1 billion bases in the course of about a week, which was quite a nice achievement. Now, the interesting thing about the MinION device on the International Space Station is that, as with all NASA technology, it was actually old technology. Now, unlike most other NASA technology, compared to other technology, it was relatively new, 6 months old. But in the MinION space, it's really old. Sorry. You can minimize that. Okay, that'll do. The space station was using a MinION version, which Oxford Nanopore have called version 7.3. Oxford Nanopore have since changed to version 9. They have also made another modification, which they haven't had at a version number 2. And a couple of weeks from now, they're bringing up an additional technology upgrade that they're calling version 9.4. So that's 3 technology upgrades in the course of about 6 months. But before I go any further, I should probably tell you a little bit about how this device does sequencing. So this device has a flow cell. This is the flow cell here, which I actually, let's see if I can. Oh. I'm not visible.
You can do
This is the flow cell. Okay. This flow cell is currently sequencing some DNA. I'll show you a bit more about that later. The flow cell has basically an input channel, an output channel, and a sequencing matrix. Now, the idea is that you've got liquid on one side, liquid on another side, and a barrier in between that things can't pass through. So a membrane barrier. And embedded into that barrier are these very tiny pores, or nanopores, which allow the passage of DNA. So in order to get DNA through the pores, there's an electrical current which is connected up to this flow cell. And the current pulls the DNA through the pores from the input channel to the output channel. Now DNA normally will shoot through the pores at about 1 million bases per second. But in the sample preparation process for preparing DNA for sequencing on this device, you attach a little, they call it a motor protein. It's actually a brake protein that binds to the DNA, sits into the pore, and it's a polymerase that slows down the movement of the DNA as it travels through the pore. Slows down the movement of the DNA to about, at the moment, 250 bases per second. At any one time, this thing can be sequencing 512 different sequences. So the potential maximum sequencing capacity of this device at the moment is whatever 512 times 250 bases per second works out as. Over the course of a 48-hour run on this device, you would expect to see somewhere between 500 megabases to 5 gigabases produced. Okay. So, as I said, NASA technology was a little bit old, running at 60 bases per second. And it actually had a 4-hour sample preparation. Now, the current technology, what I've been doing here, it's running at 250 bases per second. And I actually did the sample preparation yesterday over in building where Chris is staying. And it took me about 10 to 15 minutes to do. And as I said, expected 1 to 5 gigabases. Now, Oxford Nanopore, the Chief Technology Officer of Oxford Nanopore gave a technology update about a week ago, where he said they're bringing out this new technology, this R9.4. It's going to be running at 450 bases per second. Instead of a 10-minute sample prep, it's going to be a 5-minute sample prep. And if you add all of those together, it's going to be putting out somewhere between 5 and 15 gigabases. This is just a graph here showing the progression or the output of reads across the course of a run. And I've added to this graph R7.3, so this is what NASA was doing on the space station. And this one here, Clive Brown just had a Twitter post this morning where he said they managed to get it to 10 gigabases in one of these runs. And that's using this technology combined chemistry update together with flow cell, a pore update, which will be coming out in about 3 weeks. Additional to that, you might be interested how much one of these devices costs. if you buy these in bulk, get 48 of them, then the cost is $500 US per consumable flow cell. The device itself is $1,000 US. But when you first purchase the device, you get additional flow cells and kits to test out the device. Clive Brown said that with producing about 10 gigabases per run, it's going to reduce the sequencing costs down to about $60 US per gigabase, which puts the cost per base in similar ballpark to what Illumina is doing now with MySeq and HiSeq. Of course, Clive didn't stop there. There's additional technology that they're planning on bringing out in the next year or so. Their rapid sequencing kit at the moment will only do a single strand of DNA. So they want to get it to do RNA directly. They also want a kit which will allow you to do short fragments, also a kit for preparing samples quickly for PCR, and an Applicon kit. They also want to bring out kits which are completely dry, so that you can take those kits anywhere and you don't need to worry about putting them in a chili bin or putting them in a freezer. There are also these other devices. This device is called a MinION. Other devices which are currently being rolled out to customers called MinION. Now, Clive Brown mentioned in his technology talk that there were probably around 3,000 or so MinION devices out now. And my guess is, based on what he said, there's probably about 8 to 10 Promethean devices. Now, the Promethean devices are essentially, they're a bigger version of this, which has space for 48 flow cells. So with 48 flow cells, a slightly different flow cell, the output of these Prometheans using the current technology sequencing rate is about 1 terabase per day. And theoretically, if absolutely everything was working properly, it would be about 6 terabases per day. Oxford Nanopore is also bringing out something that they call Voltrax, which is a tiny lab on a chip device, which allows you to control fluid flow. And so that will basically allow you to pipette your sample down, hit a button, and then it'll do all the lab work for you and produce your prepared DNA at the end. Oxford Nanopore is also bringing out something called a flongle, which is something that sits in this MinION and allows you to insert another smaller consumable device into the top of that to allow for specific analytical experiments. So maybe, for example, a targeted cancer panel. And Oxford Nanopore's goal is, or their aim, is to get anyone sequencing anything, anywhere. Now, one of the biggest barriers to that is using a pipette. So they want to bring out this device which will allow someone to do sample extraction, DNA extraction, and then preparing DNA for sequencing all on a single device, which can then just be held over the top of the MinION, which will drop the sample into the MinION for sequencing, which means that all you need to do is suck up your sample, wait 5 minutes or so, hold it over the MinION, and then hit go on your sequencing application. They're also investigating technology associated with Cas9 protein, which will allow you to do enrichment and detection and possibly also fragmentation using targeted sequencing. And another technology is this thing that they're calling the smidgen, which will sit on the, I don't actually have a smartphone, here's one. We'll sit on the end of a smartphone, plug into that, and then you can do sequencing on your iPhone or your Android phone. So, a lot of interesting, cool stuff coming. So, what we want to do is to try and get out a paper before Oxford Nanopore releases their next technology update. This is going to be a bit of a struggle, given how fast they are. So we have this mouse parasite. It's called Nippostrongylus brasiliensis. It's a mouse model of human hookworm. And it's useful for people working at the Milligan Institute because it has a very specific immune response. It gives a very strong, they call it a Th2 response. Some people associate this with an allergic response, or you might call it a viral or DNA associated response. However, this parasite doesn't have a good reference genome. There is a reference genome which was generated by the Wellcome Trust Sanger Institute as part of a set of 40 parasite genomes that they were trying to sequence. This genome has a size of about 300 megabases and various statistics. The median contig length, the median assembled sequence length from short Illumina reads was about 2.6 kilobases. So, and close to 30,000 different contigs for this parasite. So this is an annotated genome which needs a little bit of work. So what we're trying to do is provide the research community with a better annotated genome of Nippostrongylus brasiliensis. So we did a run on the 7th of July this year. It was basically the first run that we were able to do once we got an R9 flow cell. And the statistics for this single run are quite similar to the statistics at a contig level for the assembled Nippostrongylus genome. We have a median sequence length of 7 kilobases, total length of 100 megabases, and 12,000 reads. What I noticed when I first looked at the reads of this run, I put them into an assembler called Canoe, and I ended up getting a contig which had a coverage of about 100 times. Now, I found this a little curious because the expected size of this genome should have been about 100 to 200 megabases. So if I've got a run which has 100 megabases of total sequence, what on earth could this contig be? So I did the first obvious thing. I loaded that contig into BLAST and ran it. And the BLAST results came back suggesting that this was a parasite mitochondrial sequence. So while I went in here trying to sequence the entirety of the Nippo genome, the first contig that came back was essentially a complete mitochondrial genome. Now, I also ran this mitochondrial genome through last to work out what parts of it map to each other. Now, we know that mitochondria are circular chromosomes. So if I have a sequence which comes out, there should be a portion of the sequence that maps to itself. And indeed, that's what I found. Here on the bottom, I have a solid line here, and here is where the sequence starts mapping back to itself. Now, what you're seeing here is the contig that came out of Canoe. So this was a contig about 20,000 base pairs in length. And what I've done here is taken those Wellcome Trust Sanger Institute reads and mapped them back to this contig. And what I discovered was that there were about 200 insertions in the Wellcome Trust reads compared to the assembled Canoe sequence. And what have we got here? 6 SNP variants. So if we ignore the indels, it's pretty close to what the mitochondrial sequence was. So I was very happy with that. This was enough for me to get started with assembling this mitochondrial genome. So that's what I did. I went through 2 additional rounds of error correction. So the first round of error correction basically got rid of all of the indel errors and the SNP errors. Second round of error correction, I think there were a couple of indels which inserted too much, so I had to put in deletions to correct. And then I circularized the genome and ran an additional error correction to fix up a few other bits. Then what I did was I remapped the MinION reads back to this assembled genome to see if there were any differences between the Sanger Institute reads and our MinION sequences that we generated from at the Milligan Institute. And what I discovered, we have sort of coverage of, say, 10 to 30 times right across the whole genome. And I found 3 variants in the genome, which were different between the Milligan Institute and the Wellcome Trust. I've just indicated here what those 3 variants are. So there's one at 5742, one at 6617, and one at 11460. End result, we got a complete mitochondrial chromosome of, I think it was 13,355 base pairs. As mitochondrial chromosomes go, that's pretty tiny. I used NCBI Smart Blast to see if I could find open reading frames, work out what potential genes they were, and ended up basically discovering and annotating everything within the mitochondrial genome, including ribosomal RNA sequences, and I used an additional program called TMScan to find tRNA sequences within the genome. Now, ordinarily, this is where you would stop doing an annotation. But I have a MinION, and it sequences using the electrical signal. So in this case, I was able to go back to the MinION reads and map the electrical signal to the genome as well. Now, when you do sequencing on the MinION, what you get out are these things called events, which are electrical descriptions of a particular section which accounts for 5 consecutive bases, pentamers. So I was able to find all the pentamers throughout the mitochondrial genome, which were identical in sequence. Then, using this electrical information, I was able to work out the sort of the median electrical signal for a particular pentamer. So then, using that information, that aggregated information, I then took my reference DNA sequence and applied that pentamer model to the DNA sequence to work out sort of an ideal signal, an ideal electrical signal for the entire mitochondrial genome. So now I've been able to annotate the mitochondrial genome with what the MinION current level should be at each base around the genome. I didn't quite stop there. The MinION samples at about 4,000 hertz, 4,000 times a second. DNA goes through the pores at 250 bases per second. So this electrical signal is actually at a sub-base level. The DNA will change slightly as it's moving through. And so I've been able to do a preliminary mapping of the sub-base signal or the raw signal and map that to the mitochondrial genome as well. And that's going to be where our paper is going to finish. Written that up, I'm just waiting for a few things before we submit. Unfortunately, I can't stop there. I see these things, I see this signal information, and I want to do more. I want to look at how this raw sub-base signal matches to the ideal sequence. So then I dug further. And I saw that the ideal signal wasn't actually the same as the electrical signal, pretty much everywhere I looked. There are some regions where it's similar, and there's a trend that it sort of follows the same shape, but the actual level isn't the same. Now, there are lots of potential explanations for this, and I can't really delve too much into all of them. But one of the things that I was curious about is, well, okay, what does this look like on a pentamer level? If I look at all of the pentamers in the mitochondrial genome and look at the raw signal level and the ideal signal level, what does that look like? And we see a reasonable correlation, but there's a lot of spread in the raw signal all across the genome. So I thought, well, maybe I've just picked the wrong points. Maybe instead of it being one base right in the middle of the pentamer, it should be the first base of the pentamer or whatever. So then I tried modifying the lag in the raw signal to see what happened when I moved the raw signal and redid the correlation. And I got this sort of interesting plot, a symmetrical plot. Now, when I was mapping the raw signal to the reads, one of the things that I did was assuming that every single base had 13 samples, 13 raw signal samples in it. So what we see here in the middle, this is mapping to the very middle of a pentamer. And this is mapping one base to the left, one base to the right, two bases to the left, two bases to the right. And what I've found interesting looking at this is just this really extreme drop in correlation as you move a couple of bases out to a sort of negative correlation, which to me seems to suggest that the signal is sort of going up and down every two bases or so. I'm not completely sure about that, but this is just thoughts that I had. I'd be welcome to discuss this with any of you afterwards to talk about why this particular image might have happened. So I sort of was interested, I saw a little bit of banding in the signal here. So just to see what that banding might mean, I concentrated on quite a dark banded area in here. So here we have, this is a very, very zoomed in version of that correlation plot that you just saw previously. Looking at, where we go? Along the bottom is the predicted ideal current for all of these pentamers. And on the y-axis is the raw signal, which was calculated at each of the locations in the mitochondrial genome where that pentamer existed. And for the most part, these ideal signals are discrete. But there are some regions where you have multiple pentamers which are coming up with essentially an identical raw signal. So, being a bit curious, I looked a bit further at this. So this one here, we have TTTTA, GTTAT, and GACTG. Now, these pentamers have an identical raw signal at that particular base position. As you go out from that position, at all of the points along the mitochondrial genome where these pentamers exist, the bases surrounding them are going to change slightly. So you would expect some change in the signal level as you stretch out. And that signal level change should increase the further away you go from that central region. And this is why Oxford Nanopore is able to base call even though you have situations where you have multiple pentamers which have the same initial signal. What they do is they capture the time sequence of signals as sequences are transitioning through the pores. And then by looking at things that change differently within a particular region, you can separate out which is the correct sequence. So here we have one base prior. We have the red sequence and the green sequence separating out, with the red and the blue together. And one base forward, we've got the red and the green sequence together, and the blue sequence separating out. So this is from the ideal signal. What about the raw signal? Well, this is where I got really confused. It's a mess. I can't really explain why this is happening. I don't understand what's going on here. Again, very pleased to discuss this with other people. One thing that I suspected might be happening is, well, maybe there was scaling issues. Maybe at a particular region where it said that it should be 90 picoamps, it was actually 100 picoamps. So one of the things that I tried was, okay, let's just assume that the raw signal at that central location is exactly what the ideal signal should be. So basically, shifting all of these lines up or down depending on what would be required to get that central location to fit in a single spot. This didn't actually help. So I've only just done this like last week or something, and I haven't really had time to analyze this further, but this is what I'm going to leave you with in terms of how deep I've tried to go with this. I've ended up with something fairly confusing, and I would appreciate any discussions on why this might be happening. So that's the end of sort of the analysis part of what I'm going to be talking about. As I mentioned previously, we do want to do a whole genome sequencing of this parasite. But there's also something else that I can show you. Let's see if this might not work. It's not working. Maybe before I do that, I'll just acknowledge people. I want to thank NZGL and Auckland University and all the wonderful people who have brought me to be here, and Oxford Nanopore Technologies and the community for allowing me to test out this device and find all these interesting new things which I never would have thought of happening, and also the people at the Malligan Institute who have helped me doing this sequencing. Okay, so now I'm going to switch out of this presentation and show you if I can find it. VLC. Here we go. Right. So I said that the MinION sequences at 250 bases per second. And I said that it produces an electrical signal. We can actually convert that electrical signal into sound using a process called frequency modulation. So here is what the very first gene in the Nippo-mitochondrial genome sounds like at 1 times speed. Okay, so that's 1 times speed. let's drop it down to 0.2 times speed, a fifth of its speed. In other words, the speed at which the sequencing would have been carried out on the International Space Station. So you're starting to hear a little bit more interesting stuff. And you sort of pick up from this the up and down variations that are happening as the bases are going through. And I'm just going to play one more. This is the first 300 bases of the mitochondrial genome, COX-1, at 10 bases per second. Okay. So none of the MinION sequencing has ever been this slow, but this is probably as slow as it would need to be for us to be able to understand and recognize particular sequences as they're being sequenced. Okay. Now, before I started the talk today, I started a sequencing run on this device. Now, this is a run in which I prepared the sample yesterday, started the run, ran the sequencing on, I think it's a laptop that Peter has now. I ran that in the lab. We disconnected it, shifted it to Chris's office, ran it again overnight. I disconnected it again, and I found out this morning that Oxford Nanopore have just released a Linux client for their sequencing software. So I installed the Linux client on my computer this morning and plugged this device into my computer and did another sequencing run. And again, just before this seminar, I've had another go. So this is about this. I mean, it's still using the same DNA that was on here. I haven't done anything to this. I haven't done any pipetting or additional preparation to this device since it was in the lab yesterday afternoon. But it's still sequencing. And this is what has been generated in the course of my talk just now. We have 1,140 sequences, about 4 megabases generated in the course of about 50 minutes.
What's the longest read?
The longest read, according to this, is between 64 kilobases and 68 kilobases. Let's see if I can get that out. What's that? Oh, I've named my laptop Gallus. This is Nippostrongylus brasiliensis. Okay, so this is a sequence that was generated at 4:56. So about 10 minutes ago. Let's pretty big. Gosh, is that all? I'm just going to run BLAST to give me an idea of how big that sequence is and what it is. Now, I'm going to map to the general nucleotide collection using, well, let's go with all the default parameters for BLAST. I think it's 100. So this will tell me how many bases it is. Yes, this is the 64 kilobase sequence that came out. So this is the longest sequence that was produced while I was giving this talk. And luckily, hopefully, this matches to the current reference genome of Nippostrongylus brasiliensis. Now, the identity scores here are a little low for what's expected for this flow cell, but I think that's probably because it's already been running for about 25 hours and hasn't had any new sample on it. If you have new fresh sample, identity from a single sequence on here is going to be about 85%. And Oxford Nanopore is continually improving their software to increase that identity. Anyway, what we end up with when we run this through BLAST is something that looks sort of very similar to what you might expect to see in IGV. Where you are mapping your short reads to a contig, except in this case, we're mapping the contigs from the reference genome onto a single read from the MinION. And so we have, over 100 contigs which are mapping to this single reference genome. So I'm hoping that this particular contig is going to help us with our Nippo assembly. And I think that's pretty much where I'll finish. So thank you for being a wonderfully attentive audience.
Thank you very much. I always reckon a true bioinformatician can take the data that works and show that. That's really cool. I know some people get away, so why don't we not have formal questions, but people can come and ask you a question now, or maybe even over a beer or something not going about. So thanks.
Okay. Do I need to stop here?